Hi, everyone. I'm Husvinee. I'm a postdoctoral researcher in Professor Brendan Candy's lab in University College Dublin. I'm currently seconded to PharmaHungary for 4 months, which is in Budapest, Hungary. It's a clinical CRO, and I'm being supervised by Dr. Zoltan Girsi. So a little bit about me is that I'm from Singapore, and I did my PhD in Singapore, and then I moved to Dublin for my first postdoc position, after which I stayed on to continue my second postdoc position. And in this case, I'm interested in identifying therapeutics for treatment of metastatic uveal melanoma. So in this project, I was lucky enough to get 2 opportunities to go on secondment. One was to Sweden, to Budapest, to do my in vivo experiments. And the other secondment is what's ongoing at the moment, which is with Crystal in PharmaHungary. So in this project, our project goal is to establish a protocol for isolating extracellular vesicles from metastatic uveal melanoma cells, because there's currently increasing evidence that shows that exosomes and their cargo can have important roles in tumorigenesis processes, as well as that they can be modulated to be used as cancer prognostic markers or to identify novel therapeutic targets and or for drug delivery. So in our lab, we are interested in identifying novel therapeutic targets and using them also on the concept of drug delivery. So in this project, what I wanted to learn was how to isolate extracellular vesicles from the metastatic uveal melanoma cells that have been treated with an anti-cancer agent, and afterwards to try to understand through proteome profiling how these metastatic uveal melanoma cells actually respond to an anti-cancer agent treatment. So the reason why we chose PharmaHungary is because they have expertise in isolating EVs, so I can actually learn a lot of methodology in terms of how to go about doing this. And I've given a list of co-authors who are involved in this project for the Eva Isolation part. So to begin with, I had to address to both of my objectives. I had to optimize the protocol, from which started with culturing these cells in a serum-free condition, and to also optimize the drug concentration required. And this was done using screening for apoptosis markers using cytoflex or flow cytometry analysis. So it was very critical that the cells were not cultured in serum and or having a high concentration of drug, because the apoptotic bodies can also be isolated as microvesicles. And further on, I had to optimize the time point for my Eva isolation. And once I decided whether it was to be 24 hours or 48 hours, then I collected the conditioned media, which was used for Eva isolation via ultracentrifugation. And the cell pellet was further stored for future use. So the isolated EVs were then used for nanoparticle tracking analysis, electron microscopy, mass spectrometry analysis, or immunoblot analysis. So far, we have actually been successful in establishing a protocol to isolate the EVs. So as my ultimate goal was to do proteome profiling from the EVs, I wanted a good working range of between 20 to 30 microgram of protein per sample. And this meant I had to optimize the number of cells that were to be seeded, as well as the volume of media required and the number of T175 flasks to be used. So what I found was that if I seeded 16 million cells, and which meant it was cells seeded in a for T175 flask, I could pool these samples and I'll have enough protein for my proteomics study, as well as for nanoparticle tracking analysis and for my EV mass spec analysis as well. So this table basically shows the EV isolation with the optimization of the number of flasks used, the cell seeding density, and those that are colored in or highlighted in blue shows the successful EV isolation methodology that was used. And this was done in watercolor as well. And once we isolate EVs, and they were supposed to be used for nanoparticle analysis. So this just gives a representative image. So what it's showing is that the vesicles that were isolated is approximately 120 nanometer in size. and they were concentrated of up to 2 million EVs, or 2 million particles per milliliter for both samples that were isolated. What was interesting was that in the ergoline, which is the anti-cancer drug treatment group that we have, there was a small percentage, approximately 30%, which had slightly larger diameter size in comparison to the majority of the particles that were isolated. So this indicates that the anti-cancer drug may be changing the EV profile slightly in these cells. And the second part of my objectives was to isolate once the EVs were isolated, was to perform a proteomics analysis. So in this case, I had to optimize the protocol that I was going to use. So we did a trial round whereby we used one set of samples were just vacuum dried and then post-processed. And then one sample was prepared with PBS that was either TCA precipitated or post-processed. And the last one was prepared using proteomics kit. And through this trial run of our mass spec analysis, we found that the vacuum dried samples gave the highest number of IDs of 573, and the proteomics kit gave us the lowest number of IDs. So we have now decided to go with the vacuum dried sample processing method for our mass spec analysis. So this is the diagram that basically shows the common number of protein IDs that's being picked up across the three different techniques used, as well as with the vacuum dried samples, we have an addition of 42 IDs, which is not overlapping with either the PBS method or the proteomics method. So that's the overall project objectives which I've achieved so far. So the samples have been collected and they are now being shipped to Dublin, where we will do the mass spec analysis. So from this secondment, they have definitely learned a lot in terms of going about optimizing a protocol for Eva Isolation, so that this standardized protocol can be used in future in our lab for isolating EVs from these cells. Also, by optimizing the protocol for proteome profiling is actually giving really good, because this has not been done not only in our lab, but also in PharmaHungary. So in future, this protocol may be used by them for their future studies as well. So this has actually helped to strengthen our collaborative network. We are currently working on co-authoring a review article, as well as we are planning to publish our results that we have obtained from our EV isolation. I have gained a lot of experience working in a CRO, not only in terms of project management, but also being exposed to different work culture in terms coming from an academic background. And during this time, I was also able to disseminate my findings in terms of contributing to school talks, as well as IOBA meeting with the project manager. And this work will be presented in an international conference, as well as for working on raising awareness on uveal melanoma. And personally, this has given me a better idea on the types of career track I would like to take part in the future. So with that, thank you for having me, and I definitely have to thank Brendan and Yolanda for giving me this opportunity to go on a secondment. Thank you.